Good morning. Thanks everyone for joining us. Irina, you want to take it away?
Thank you. Yes, so thank you everybody for joining us for today's Distinguished Seminar Series presentation. Just as a reminder, the Distinguished Seminar Series features presentations by outstanding thinkers and scientists sponsored by the Allen Institute for Brain Science. Distinguished speakers are selected based on the impact of their interdisciplinary research to the neuroscience community. So our today's speaker, Dr. Addy, it's a great pleasure to have him here speak today. Dr. Addy is an associate professor of psychiatry and of cellular and molecular physiology at Yale School of Medicine. He received his bachelor from Duke University in biology and his PhD from Yale University in neuroscience, studying the role of calcineurin in nicotine-mediated changes in behavior and neural activity under the guidance of Marina Picciotto. Dr. Addy is a prolific scientist and a community educator. His research uses latest techniques in electrochemistry and electrophysiology in combination with rodent behavior to understand the role of neuromodulators like acetylcholine and dopamine, as well as L-type calcium channels in substance abuse and mood disorders. His work is essential in understanding the neurobiological mechanisms of addiction and of psychiatric illnesses overall. He also is active in diversity, equity, and inclusion work at Yale, serving as an inaugural director of Scientists, Diversity and Inclusion, as well as Minority Organization for Retention and Expansion at Yale. So he helps train faculty, postdocs, and graduate students in this area. He also hosts a podcast called Addy Hour, where he invites speakers and discusses topics related to neuroscience, social justice, mental health, and faith. I highly recommend checking it out. So in addition to being a brilliant scientist, he also is an excellent communicator and a deep thinker. And it's a great pleasure to have him speak today. So please join me in welcoming Dr. Addy.
Thank you so much for that introduction. Very humbling. I definitely appreciate the introduction, and going all the way back to my grad training too. It's been a while since I've thought about some of those studies, so it's nice to be reminded of that in a sense. It's great to be here with all of you this morning for you all, afternoon for me, and morning or afternoon depending on where people are tuning in from. Definitely appreciate the invitation, both because of what the Institute stands for and some of the big science and the ways that you all think about things, but also because of the distinguished speakers that have been in this obviously distinguished lecture series in the past. So it's really an honor to be included in that group as well. So as you mentioned, I'm going to talk about today some of the work that we've been doing over the last few years, thinking a lot about substance use and mood disorders, thinking about the basic neurobiology of what allows those to emerge in the first place, and also thinking about ways to try and have some therapeutic interventions as we understand the underlying neurobiology. I'm especially interested and excited to talk to this group, given what you all think about and the ways that you all go about science. I'm sure there are many things I can learn from you all as well in terms of perspectives and even potential collaborations and ways to think about things coming up down the road as well. So let me do the classic share screen, and then we will jump in. I also mentioned I'm going to time myself as well, because in Zoom land, it's very easy to lose track of time without seeing all of your faces in front of me. So I thought it would also be good to just pay attention to that. And I definitely want to leave room for time for questions at the end as well. So again, today I'm going to be focusing on some of our foundational work, but then also some of our more recent work, looking specifically at L-type calcium channel regulation of dopamine, and looking at that in the context of motivated behavior. Again, in our preclinical models of both substance use and mood disorders. And so at the beginning of my talks, I always like to give a framework for what we really are trying to understand from a global standpoint. I think this is particularly germane to this audience, especially the work that is done at the Allen Institute as well. And again, I'll be really curious to hear thoughts about some of these topics as we go through the talk as well. But from a very global standpoint, we're really just interested in trying to understand the neurobiological basis of complex behavior. Now, of course, within our work, we're doing that specifically within the context of both substance use disorders and mood disorders, and thinking about behaviors that are associated with both of those illnesses in particular. Now, obviously, you all are a sophisticated audience, and so these are things that you've likely thought about before, but oftentimes when I'm giving talks in public as well, I'll just have audience members share with me some of the behaviors that come to mind when they think about substance use disorders or when they think about mood disorders. And it's always really insightful for me to hear the types of things that people share and the insights that they have into some of these things already. And so for us, we're really just trying to look at some of those same behaviors and think about, well, what is actually allowing these behaviors to emerge in the first place? And so for our work, we've really decided to zoom in and focus on motivated behaviors in particular, and thinking about things which an organism might seek out that are beneficial for the organism, or things that we might, as an organism, avoid that are harmful. And again, here we're really trying to understand the neurobiological basis, neurobiological mechanisms that allow these behaviors to emerge in the first place, and then thinking about those behaviors that are associated with these illnesses. As a second goal, we're hopeful that by identifying some of those neurobiological processes, we might actually be able to gain some insight into the etiology of the illnesses in the first place. And then as a third aim, we're hopeful that by identifying these processes, we can actually also move things forward and identify some of the novel therapeutic interventions that we may be able to develop by targeting these processes in particular. So for today's talk, I'll be focusing on our research in two different aspects. First, I'll go through our work looking at positively motivated behavior, particularly in the context of substance use disorders, so looking at things like reward taking and reward seeking. And then for the second half of the talk, I'll shift to some of our more recent work looking at negatively motivated behavior, particularly within the context of mood disorders. So thinking about behavioral responses to stress, anxiety, anhedonia, avoidance, and amotivation. And as you likely noticed in my cartoon image in the top, I've also drawn arrows between these two intentionally. And one of the reasons is because we're also starting to think about comorbidity quite a bit as well. So as many of you may know, those struggling with substance use disorders have a much higher prevalence for mood disorders and vice versa. So starting to think about the common underlying neurobiology that might facilitate both of these types of illnesses, also facilitate the comorbidity. And then again, to think about therapeutic interventions that might actually address both of these simultaneously. So just to give a little bit of background about our area of focus, within the substance use disorder realm, we've actually decided to focus on cue-induced craving and relapse. And our main reason for doing so is because this is such a major factor that often prevents individuals from having long periods of abstaining from drug use if that's what they're seeking to do, and also from abuse of those substances as well. This, of course, is based on stimulus reward learning, so going all the way back to freshman psychology and thinking about associations between unconditioned stimuli and conditioned stimuli. So I've given a few different examples. On the left here, we have an animal in the wild that has learned, of course, to associate the availability of food with some of these discrete cues in the environment. We have a cue that's very salient to me, which is a coffee maker. I had my coffee this morning. For me, for many others, perhaps this conglomeration of metal and plastic, when you see it, elicits a sense of craving. For those who are struggling with substance use disorder, that might be drug paraphernalia or context or people that they're used to taking those substances with. So again, going back to our freshman psychology, thinking about the unconditioned stimulus being the reward, that's repeatedly paired with a conditioned stimulus, so much so that that conditioned stimulus can elicit craving and promote reward-seeking behavior. And I always emphasize here that this is important, as I've mentioned, both for natural rewards and for drugs of abuse. So I've tried to highlight that in these images as well. I think it's also worth mentioning that these conditioned stimuli themselves can also gain incentive value where they become desired or wanted. So this is work that's been pioneered over the last 15 years by Kent Berridge and also Terry Robinson and others at Michigan and at other places as well. But again, this idea that the conditioned stimulus can become desired and wanted. I think a helpful example here is to think of someone who has been a smoker for years and has quit smoking. You know, talking to a lot of these individuals, they'll say that they often will miss the throat hit they used to get every time they smoked a cigarette. Now, that throat hit itself doesn't have anything to do with the reinforcing effects of nicotine or the euphoria or the buzz that someone gets from smoking a cigarette, but it becomes so desired that when someone has abstained from smoking for quite a while, they have a craving and desire for that conditioned stimulus itself, suggesting that conditioned stimulus actually has an incentive value to them as well. So with that framework, about 12 years ago when we started the lab, we were trying to think about some of the areas where we could focus within brain systems to think about what might be underlying this stimulus reward learning. So at the time, we went back about a decade or so. Now this is about 2 decades old, but looking at some of the pioneering work that had been done by Anna Ray Childress and colleagues on specifically using imaging in cocaine-dependent individuals. What they did in these experiments was to show these individuals either a nature-related video or a cocaine-related video that had cocaine cues associated with it. And they saw increased activity in specific dopaminergic regions of the brain. So in this study, they're showing both in the amygdala and in the anterior cingulate, they saw increased activity when the individuals were shown cocaine-related videos. They went on and carried out this work with different types of substances and also saw effects in different regions as well. So looking at things like cocaine, as I mentioned here, but also alcohol and opioids, and saw increased activity in several dopaminergic regions, including the prefrontal cortex, the striatum, and the amygdala. So this gave us in the field and our lab in particular, at least some insight into some of the regions that may be involved in processing these cues. But we were also informed by a lot of the preclinical literature. So obviously I'm showing a human sagittal brain here, but just want to emphasize that for our work, we decided to focus both on the dopamine cell body region of VTA and on dopamine terminals in the nucleus accumbens. And there were a couple of different reasons that we decided to do that. So again, going back to the preclinical literature, this is pioneering work that had been done by Wolfram Schultz and colleagues, which many of you are likely familiar with. This was work that was done in macaque monkeys, basically looking at the activity of dopamine neurons in the midbrain in response to an unexpected reward. So what they did, what they found when they used physiology was an increase in burst firing of these dopamine neurons in response to this unexpected reward. So I should mention that these dopamine neurons are known to fire in 2 different modes, either burst firing with high frequency, irregular firing frequencies, or tonic frequencies, which are much lower frequency, more like a pacemaker. And so in the raster plot, as you can see, they have a dramatic increase in burst firing when the monkeys got an unexpected juice reward. They then performed Pavlovian conditioning in these animals where there was a conditioned stimulus that always predicted the availability of the reward. And what they found was a shift in the dopaminergic response.
Sounds like we might have lost me, so we will be right back. Thanks for your patience, everyone. We're working on getting me back online here. As a reminder, while we wait for Need to get reconnected, if you do have questions at any point during today's webinar, you can use the Q&A box in Zoom or the chat on YouTube to ask those questions. It looks like Nee is back online.
So I think Zoom froze on me, apparently.
It happens. So looks like we lost you around the basic dopamine role and reward processing slide.
Okay. Well, I'm glad to be back. My pointer started moving very slowly and I was getting a little concerned, and all of a sudden I was gone. So let's make a second attempt here. All right, does this look familiar?
Is still loading.
Oh, okay, let's try again.
Yep, that's the slide where we lost you.
Okay, excellent. Apologies for that. I guess we're keeping things a little bit interesting today. All right, so we'll try and pick back up. So I think I was starting to talk about the phasic dopamine activity. So specifically that these dopamine neurons can fire with rapid burst firing like activity anywhere from 20 hertz or higher. They also show tonic-like activity or pacemaker-like activity at times. And what the investigators found is that when they presented an unexpected reward to the macaque monkeys, they got a dramatic increase of burst firing within the midbrain of these monkeys. And so showing that here in their raster plot. And just before I go ahead, I'm going to put my timer back on as well. And then what they did was to now perform Pavlovian conditioning, where they actually presented a conditioned stimulus that now always predicted the availability of the reward. And so what they found is there was actually a shift in that dopaminergic activity. So now they had an increase in burst firing, which they observed in response to the conditioned stimulus, not much of a change in response to the reward itself. They did also go on to look at what happens when an expected reward is removed or not presented, and looked at things like reward prediction error. But for the purposes of today's talk, I'm going to focus primarily on the responses to reward and to the conditioned stimuli. Again, emphasizing the fact that this dopaminergic system is very dynamic and can respond to both of these and shift in over time. Now, this again was focused on the midbrain area, but people were also curious about what's happening at the level of the nucleus accumbens, where dopamine is released, can bind to postsynaptic receptors, and actually influence goal-directed behavior through intersections with the limbic system. So these experiments were actually done in the laboratory of Dr. Mark Whiteman and Dr. Regina Corelli, who were collaborating, performed by Jeremy Day, who was a graduate student at the time in the Corelli lab. And basically what he was doing was using voltammetry to now look at changes in dopamine concentration in the accumbens in a very similar task. This was performed in rats. So what he did was to present the rats with an unexpected reward. You now see a dramatic increase in dopamine concentration in response to reward, and not much of a change elsewhere in the trace. If they then again perform Pavlovian conditioning in these animals, they also see an increase in response to the conditioned cue, and not much of a response in terms of concentration to the reward. So again, highlighting the dynamic nature of the dopamine system, both in terms of its firing properties and also release in nucleus accumbens to respond either to unexpected rewards and to cues that predict rewards. So again, a lot of this showing nice correlated behavior in terms of how the brain processes these cues. One thing that we became curious in was what actually regulates the dopaminergic activity itself. There was a lot of evidence showing that acetylcholine played a key role in regulating dopaminergic activity. There was a paper that came out in 2008 from Uwe Maskos, a review paper that called acetylcholine the neglected master modulator of dopamine signaling. So we were intrigued by that. Obviously, I had some background in acetylcholine signaling from my grad training. And we decided to actually look at some of these processes and mechanisms within the context of these motivated behaviors. So again, thinking about substance use disorder, we of course need a model with which to test these types of questions. So we turned to some standard behaviors that have been used in the field for a while. They're still used, and that being IV drug self-administration, which again, many of you may be familiar with. So just to briefly go through, basically we have Sprague-Dowdy rats that we're using. We perform surgical manipulations where we insert a catheter into the jugular vein. The animals recover, and we allow them to then enter an operant box to press a lever to receive intravenous drug, which gives very rapid access to the brain. So in this chamber, the animal can actually press either the active lever here, which will deliver cocaine, combined with a 6-second auditory and stimulus cue, which becomes a conditioned stimulus. There's also an inactive lever. If they press that lever, there's no programmed consequence. What we see over time is the animals get very good at learning to distinguish between the two levers. You can see over a 10-day period, they have a pretty good rate of responding on the active lever that produces cocaine. And they are pretty consistent in their lever pressing as well, not much pressing on the inactive lever. After this 10-day training period, we take the animals through a period of forced abstinence, which is similar to what we're trying to model with detoxification. They don't have any access to the cues or the drug. After that abstinence period, we place them back into the operant chamber and again allow them to press either the active or inactive levers. This time, if they press the active lever, they don't receive cocaine. They only get the cue that was previously associated with cocaine. Again, if they press the inactive lever, there's no programmed consequence. And we can see if we look over different periods of time, anywhere from 1 day of abstinence out to 45 days of abstinence, we can see they press many more times on the active lever compared to the inactive lever. And this really is due to the association between the cues and the drug. If you do the same experiment, but present the cues randomly during a training and don't pair them together, you don't see this behavior emerge during these abstinent days. Similarly, if you take them through abstinence but don't present the cue when they press the active lever, you don't see this behavior emerge. So it's really this tie between the cue and the drug that allows this to emerge, and we use this as our metric of cue-induced cocaine-seeking behavior. So with these tools in mind, we did a whole bunch of work specifically looking at the dopaminergic system and the acetylcholine interactions to see if that would influence the behavior. I'm just going to summarize some of that pictorially here, but basically within the VTA to nucleus accumbens system, we saw if we stimulated these dopamine neurons with a burst-like stimulation pattern, we could enhance that drug-seeking behavior. If we blocked dopaminergic activity, we could decrease or block that behavior from emerging. Similarly, if we block dopamine receptors, D1 receptors specifically, that are preferentially activated by phasic dopamine release, we again could block the behavior. If we enhance acetylcholine tone, which would also facilitate dopamine release, we could enhance the behavior. So we could move it in different directions. And we also started to look at specific receptor mechanisms that were important. So both nicotinic and muscarinic receptors within the system, if we block those, we again were able to block the behavior and also decrease dopamine signaling. So basically, just summarizing here, we wanted to see if that was something that was specific to our drugs of abuse or also much more global. Because as I mentioned before, these processes are also important for natural reward seeking. What we found is that if we block nicotinic receptors, we specifically attenuated drug-seeking behavior, but did not alter food-seeking behavior. In contrast, if we block muscarinic receptors, we were able to manipulate or modulate both drug-seeking behavior, cue-induced drug-seeking behavior, and cue-induced behavior for natural rewards. So there seemed to be a distinction there. The nicotinic component seemed to be specific for substances that are often used in abuse, whereas the muscarinic system was more global, both for drugs and for natural reward seeking. So before I shift and talk about some of our work, specifically with L-type calcium channels, I also wanted to mention a little bit of work that we did to start to think about the pathways that might be involved in terms of circuitry as well. Obviously, this is nowhere near the scale of a lot of the work that you all do across the entire brain, but we want to at least try and start to dissect some of these specific pathways and think about how they may work in the context of the entire brain. So there is a lot of evidence showing that input to the VTA, cholinergic input in particular, comes from the mesopontine area, the lateral-dorsal tegmental nucleus. And so what we want to do initially, before looking at the specific cholinergic input, was to see whether this input from the LETG to the VTA actually influenced dopamine signaling within the nucleus accumbens. So my postdoctoral fellow at the time, Wojciech Selecki, performed these experiments, infused channel rhodopsin specifically into the lateral dorsal tegmental area, lateral dorsal tegmental nucleus, put his optical fiber in the VTA to specifically stimulate terminals from the LETG into the VTA, and then put his recording electrode with voltammetry in nucleus accumbens, and indeed was able to stimulate dopamine release in the nucleus accumbens by specifically activating lateral dorsal tegmental region terminals within the VTA. So again, validating and supporting this functional connectivity. And now we can move forward and actually start to think about whether that input is facilitating some of these behaviors. And we can use some of the genetic manipulations in rats to specifically look at these cholinergic inputs and see if they are regulating the behavior as well. But finally, getting back to what I mentioned in the title, we've also been curious about other mechanisms that can regulate some of this dopaminergic activity. And for that, we actually have formed a collaboration with Anjali Rajadasha at Weill Cornell Medicine to specifically look at the L-type calcium channels. And there are a few reasons that we decided to do that. One is because there is a story, and there has been a story for quite a few years, showing that L-type calcium channels do play a specific and important role in regulating dopaminergic activity. So in this study in particular, the investigators found that if they activate the L-type calcium channels themselves, that is sufficient to induce burst firing in these dopamine neurons. That was also intriguing to us because we knew that just stimulating the cholinergic receptors themselves using carbachol was also sufficient to induce burst firing. And we had been wondering if there was an interaction there as well. As you can see, as I've highlighted here, just one potential, thinking about alpha-6 containing nicotinic receptors within the postsynaptic membrane in particular, which are permeable both to sodium and calcium and could potentially recruit and activate the voltage-sensitive L-type calcium channels. So that's something we've been thinking about. It was interesting to also look in literature and to see that there was an interaction that seemed to occur with this cholinergic receptor mediated burst firing. So I mentioned if you stimulate the cholinergic receptors with carbachol, which is non-selective and activates the cholinergic receptors, you can induce burst firing. That is actually dependent upon L-type calcium channels. So if you block L-type calcium channels with nifedipine, you lose that ability to be able to have this cholinergic induced burst firing. So we became very curious about this intersection, this interaction, and we actually just wanted to ask the first very simple question of whether these L-type calcium channels may also regulate behaviors that are mediated by dopaminergic activity. So for these experiments, what we did was basically to take our animals through cocaine self-administration, take them through 10 days of abstinence, and then look at their cue-induced cocaine seeking using the behaviors that I described earlier. What we did before that test was basically just to infuse L-type calcium channel antagonists directly into the VTA and to see how that impacted the behavior. What we found in the first experiment was that indeed, if we blocked L-type calcium channels within the VTA, we could dose-dependently decrease cocaine-seeking behavior. So as we can see here, these are animals that went through cocaine self-administration and abstinence. They had vehicle infused into the VTA. You see they pressed many more times for the active lever compared to the inactive lever. As we increase the dose of isradipine, we see a decrease in that activity where they are pressing much less for the active lever that produces a cue compared to the control animals. We also performed these experiments with a different dihydropyridine, nifedipine, and found very similar effects. So we felt very confident that the L-type blockade was indeed decreasing this behavior. But again, we are curious, is this something that's specific to cocaine or is this more generalizable to natural rewards as well? So we took another subset of animals through sucrose self-administration, took them through 10 days of abstinence, and then asked the same question. What happens if we block L-type calcium channels in the VTA? Does that affect their cue-induced sucrose-seeking behavior? The answer was no. So again, this is very similar to what we saw with nicotinic receptor blockade, where the effects seem to be specific to cocaine and did not extend to cue-induced natural reward-seeking behavior. So we were curious whether, again, this could be mediated by dopamine signaling within the VTA to accumbens pathway. As I mentioned to you before, we had found that all of our manipulations that decrease the behavior also decrease dopamine signaling. So that's what we predicted we would find here as well. So in this experiment, this was performed now in cocaine-abstinent animals. We have a stimulating electrode in the VTA to stimulate burst-like activity in dopamine neurons and to be able to measure dopamine release in the accumbens. We also coupled this with a microinjector where we could infuse specific pharmacological agents directly into the VTA to see how that influenced dopamine signaling in these cocaine-abstinent animals. So again, we're predicting if we block the L-type calcium channel, we'll also decrease dopamine signaling. What we saw was the exact opposite. So here in a cocaine-abstinent animal, we have our stimulation here. We have our vehicle animal, our increase in dopamine. If we block L-type calcium channels with isradipine, we actually enhance the response. So completely opposite of what we predicted, and we weren't exactly sure why or how that was happening. But as the next step, we decided to at least look in our sucrose-seeking animals where we knew there was no behavioral effect of isradipine. It turns out there was also no electrochemical effect of isradipine on dopamine signaling. So even though this was opposite of what we predicted, it was very consistent in terms of correlating to our behavior. So at this point, we were still a little bit confused, but we decided to at least look and see whether there was a difference in maximal evoked dopamine release that might have been happening at baseline before we infused the isradipine to block the L-type calcium channels. It turns out there was not. So in our naive animals, in our sucrose-abstinent animals, in our cocaine-abstinent animals, there was no change in the maximally evoked dopamine release. So it's something that seemed to emerge when we blocked the L-type calcium channels specifically. So at this point, to be honest, we were still very confused and couldn't really understand why we were seeing these effects, especially based on what we had seen before with our cholinergic manipulations. But we were also thinking therapeutically in terms of trying to move things forward and knew that if this was going to be translatable, we'd also need to be able to do this, at least at this point, with some type of systemic administration. So what we decided to do next was to just ask that question. What if we give systemic administration of isradipine? How does that affect cocaine seeking and dopamine signaling? I should also mention that this isradipine that we're using is actually an FDA-approved medication for hypertension, acting at L-type calcium channels throughout the body. So we've also been thinking about whether this might be something that could move things forward in terms of repurposing. So what we did here was to repeat the same experiment, except to give IP administration of the L-type calcium channel antagonist 15 minutes before the test, just to allow it enough time to access the brain. We see effects that are very similar to what we see with the VTA infused isradipine, and that we can decrease cocaine-seeking behavior. We also want to look at the dopamine signaling. So again, using voltammetry and looking at dopamine signaling after the systemic administration. And again, what we saw is an increase in dopamine signaling in the accumbens when we block L-type calcium channels, this time with systemic isradipine administration. Incidentally, that increase starts about 15 minutes after injection. So it seems our behavioral timing was actually pretty good in terms of our estimation. At this point, to be honest, we were still a little bit stuck with our original hypothesis and trying to think if there might be some way that there were perhaps changes in tonic dopamine that were much more than what were happening with phasic dopamine, and perhaps we were still seeing some type of overall decrease in activity. So we were too stuck on our original hypothesis, but we decided to look at what happens if we stimulated the VTA with a tonic-like stimulation pattern. But we saw effects that were very similar to what we found with our burst-like stimulation. So at this point, we decided, okay, the data is the data. It's showing us that there is actually an elevation in dopamine signaling when we block the L-type calcium channels, even though that's opposite of what we predicted. We finally decided to ask the question, is that increase in dopamine actually facilitating this change in behavior? So for that, we wanted to see, well, what if we block dopamine receptors in the nucleus accumbens when we've given systemic isradipine? Does that somehow remove the effect of systemic isradipine? That was a little bit of a difficult question to address because as we knew from the literature, blocking those dopamine receptors in the accumbens in and of itself will decrease the cocaine-seeking behavior because those dopamine receptors are involved in that behavior. So showing here are experiments with the dopamine receptor antagonist, non-selective, which blocks both D1 and D2, flupentixol, we see a dose-dependent decrease in cocaine-seeking behavior when we infuse this directly into the accumbens. So what we decided to do was to use a sub-maximal dose of the flupentixol and to see whether we could use that to, again, investigate this question of whether these dopamine receptors are actually involved. So we only got modest decreases in cocaine-seeking with this 7 microgram dose of flupentixol. So what we decided to do next was to combine this with our systemic isradipine and to see whether these dopamine receptors were involved. So just to walk through this slide here, again, all these animals went through Q-induced cocaine seeking, and they all went through the same abstinence period, the 10-day abstinence period. These are the animals that got vehicle IP administration and vehicle into nucleus accumbens. You see dramatic and robust Q-induced cocaine-seeking behavior. If we block L-type calcium channels with systemic isradipine, we decrease that response. If we give the submaximal dopaminergic antagonist, we get a modest decrease. But if we give both of these together, so we have the submaximal flupentixol and then give isradipine, we're no longer able to decrease the cocaine-seeking behavior. In comparison, we see much higher behavior compared to when we give the isradipine alone. So suggesting that this change in behavior with isradipine was actually due to an increase in dopaminergic activity that was then acting at dopamine receptors in the nucleus accumbens. So again, bringing us into a broader picture, suggesting that we could actually manipulate behavior either by increasing dopaminergic tone or by decreasing dopaminergic tone. So increasing it with isradipine, decreasing it with some of our nicotinic and muscarinic receptor manipulations. So something that we were surprised by, but showed us again the dynamic nature of the system. And as we thought back to some of the literature, there is evidence both from the preclinical literature and from the clinical literature showing deficits in dopamine signaling during drug abstinence. So perhaps when we elevated that dopamine with our isradipine, that was one way to be able to alleviate some of those behavioral responses. Even though there are dopamine deficits, there's also evidence showing that you still have dramatic increases in dopamine in response to cues. So perhaps our muscarinic and nicotinic receptor manipulations are decreasing those responses to cues and again can get to the same outcome in terms of decreasing the cocaine-seeking behavior. So it showed us some of these potential mechanisms that might be in place. And one of the things that we're trying to do moving forward is actually to collaborate with our clinical colleagues to see whether some of these L-type calcium channel inhibitors, again, they're FDA approved, may also decrease craving in humans who are dependent on cocaine, and whether that might actually be acting through the dopamine system as well. So we're in the process of actually setting up some PET imaging studies to do exactly that. But again, I'll be curious to hear feedback from you all as well, just thinking about this on a larger scale and different ways that we can think about other aspects of brain circuitry and pathways that may also be contributing to some of these, and thinking about how that will have effects across the brain as well. And just to keep an eye on time as well, but I am going to shift gears and now talk a little bit about some of our mood disorder related work and then tie these all back in together at the end. So again, going back to one of the diagrams that I showed earlier on in the talk, we still have been thinking about this VTA to nucleus accumbens intersection. And as we were starting to think about some of these aspects of substance use disorder, we were also impacted and intrigued by two papers that came out in 2013, one from Kay Tai, who I know was also a speaker in the series previously, but basically showing the causal relationship of activity within the VTA to accumbens circuitry in terms of responses to stress and anxiety. So a paper that Kay Tai published in 2013, also a paper by Chaudhry and colleagues from the Han lab in 2013, basically looking at very different types of behaviors, but all showing causal relationships. So again, for us, the question for us was, if the dopaminergic system is playing a causal role in some of these behavioral responses to stress, what about acetylcholine that's actually regulating the dopamine system? That again might be playing a role in some of these responses as well. But kind of going back to that theme of acetylcholine being a neglected master modulator, there weren't any studies that had looked at midbrain acetylcholine in terms of how it may actually be impacting responses to stress and anxiety. But that's not to say that people hadn't thought about acetylcholine in terms of mood disorders before, especially within depression. This is a hypothesis that has gone back about 40 years or so, was first proposed by Dr. Janowski and colleagues based on previous work that had been done, specifically with observational work looking at individuals who had been exposed to certain organophosphates and pesticides that elevated acetylcholine's levels in the brain. And people had noticed there was a lot of increased depressed mood associated with some of that. So Dr. Janowski and colleagues actually proposed this cholinergic hypothesis of depression based on some of these observations, again, and specifically looking at physostigmine, acetylcholinesterase inhibitor, and noticing that people who were exposed to physostigmine showed things like increased depressed mood, lethargy, slowed thoughts, apathy, decreased energy, and psychomotor retardation. So there had been work over the years looking at different aspects of cholinergic components that might impact some of these responses. So again, looking specifically at nucleus accumbens, work that was done in the lab of Paul Gringard, looking at the hippocampus, work that was done by Marina Pachoda, and also looking at prefrontal cortex, work that was done by Ron Duman, specifically looking at the rapid antidepressant effects of scopolamine. But again, no work that had looked at acetylcholine within the midbrain. So that's in fact where we decided to start. And just to summarize some of that work, we did very simple experiments early on just to show that if you increase acetylcholine tone within the VTA itself, that is sufficient to induce pro-depressive, anxiogenic, and anhedonic-like properties with tests like the forced swim test, the Elevate Plus maze, and also the sucrose preference tests. Now, of course, in terms of therapeutic interventions, we don't want to exacerbate some of these symptoms. So we're also trying to think about, well, how can we block or potentially attenuate some of these responses? So for this work, we've been looking particularly to think about what specific receptors or pathways within the circuit we may be able to block or manipulate to decrease some of these responses. And for some of this work, we've actually been looking specifically at M5 receptors, which are known to be localized on dopamine neurons within the VTA and also at terminals in the nucleus accumbens. Now, the muscarinic receptor field has been hampered for years by a lack of subtype selective compounds. But we've been fortunate to collaborate with Jeff Kahn and his colleagues at Vanderbilt University, they've been pioneers in developing a lot of these specific compounds over the years. So basically, we did some work looking at some of those components, specifically with the behaviors I mentioned before. So things like the forced swim test, Elevate Plus maze, and sucrose preference test. And we found a role for M5, which seems to be mediating some of those processes. But we've also been trying to think more broadly about some of the symptoms associated with some of the mood disorders and thinking about if there are other behaviors that we can look at that might have even greater translational applicability. So one of the behaviors that we have turned to is the effort-related choice paradigm, which I wanted to go through here for those who may not be as familiar with it. Basically, what this does is a way to look at aspects of motivation and amotivation in these animal models. So basically, in this situation, the animals are placed in an operant chamber and they have two food options. One, they can acquire freely available lab chow. Alternatively, they can press a lever 5 times to receive another type of chow, which is much more palatable to them and much more desired. So basically, they're given options for a low effort low reward availability or high effort, high reward option. And you can train the animals over time to get to a point where they'll take a certain amount of the free chow and then also work up to a certain extent for the highly preferred reward. And what John Salomon and his colleagues have demonstrated over the years, that you can also manipulate this motivation by manipulating the dopamine system. So they've shown very nicely that if you actually deplete dopamine or you use dopaminergic antagonists, the animals will not work as hard for the high preferred reward. They're much more likely to take the freely available chow. In contrast, if you enhance the dopamine system, you'll actually see a shift where they'll spend much more effort with the much more effort on the FR5 schedule to actually obtain the highly preferred reward. Now, the really nice thing about this task for us is there's actually a human correlate to the task as well that was developed by Michael Treadway and colleagues. And this is called the human effort expenditure for reward task. Basically, what they'll do is something very similar. This time that individuals will actually work for monetary reward. They'll be able to get a low monetary reward by being able to press the mouse with their dominant finger to get that reward a certain number of times, or they can use their non-dominant pinky to get a higher value monetary reward. And what Michael Treadway and others have found is that individuals with major depressive disorder or who are struggling with anhedonia show a decrease in the reward sensitivity, so much more likely to take the lower value reward and less likely to work for the high value reward. The Treadway lab and others have also shown that, again, if you increase dopaminergic activity, you can also increase willingness to work for the reward. So here we have a nice correlate of the preclinical behavioral outcome that actually matches what we see in humans as well, and matches well onto those who may be navigating major depressive disorder or other mood disorders. So for us, what we want to do moving forward was to ask the question, well, we've seen that we can manipulate the cholinergic system and adjust and change responses with some of these things like the forced swim test, LA plus maze, sucrose preference, some of the classical behaviors that people have used. But do we also see changes in motivation if we look specifically at effort-related behavior? So this is data that's hot off the press, or not hot off the press yet, hot off of the data collection that we're actually putting into publication, but I wanted to share it with you all here as well. And basically what I'm showing you is in the females at first. So with our chow, we actually don't see that many changes in their behavioral responses. So again, this is a freely available chow. We're again using physostigmine here. So if we give physostigmine directly into the VTA, we see no change in the chow. If we use a negative allosteric modulator of the M5 receptors, we see no change, and no change with both together. That's different than what we see if we look at the high effort reward seeking and obtaining behavior on that FR5 schedule. So specifically looking at the lever pressing behavior. And what we see is that if we increase acetylcholine tone, we actually decrease their motivation. So they're not working as hard for that high effort reward. We don't see any change with a negative allosteric modulator of the M5 receptor themselves. But if we put these two together, we see that if we block the M5 receptors with a negative allosteric modulator, we are no longer have an effect of physostigmine to decrease that response. So in effect, we're reversing the effects of physostigmine by blocking the M5 receptors. And we see something very similar in the males as well. So again, no change in response to the chow consumption. But we do see, again, if we increase acetylcholine tone, a decrease in their lever pressing, no change in the negative allosteric modulator itself. But surprisingly, when we put both together, we actually get an increase in the lever pressing, much more than we see at baseline. So our next step here is to actually do some more dose response analyses to see what may be allowing this to occur both in the males and the females, and to try and understand why we actually have such a dramatic shift where the males will actually spend much more time on the high effort task than they did at baseline. And we're also going to look at muscarinic receptor expression specifically within these pathways to see if there may be differences in receptor expression between the males and the females that may be facilitating these effects as well. But again, tying it back to the L-type calcium channels, we're also starting to think specifically about how L-type calcium channels may also be important in some of these behaviors which we know are regulated by dopaminergic activity. So just to highlight some of the work that our collaborators had done initially in this area, this is work again that was done in the Rajadakshina lab by a postdoc at the time, Arlene Martinez Rivera. And what she did here was actually to look at, again, L-type calcium channels in the VTA to see how they may be influencing responses to behaviors that are implicated in mood disorders. And what she found is if she specifically activated receptors within the VTA, she actually got a decrease in sucrose preference test, so suggesting an anhedonic-like response. She also saw an increase in mobility time in the forced swim test, suggestive of a depressive-like response. And then in social interaction, saw a loss of social interaction, again, if she activated those receptors within the VTA. So all going in the same direction as what we saw with our physostigmine. But again, one of the things that we want to think about therapeutically is how can we reverse some of these responses and effects? So to tie things together, and before I wrap up and go to questions, I also do want to mention how we've been thinking about things, both in terms, again, of comorbidity between substance use disorders and mood disorders. One of the things that we have tried to establish and observe in our animal models is to see whether that comorbidity can be examined in our animal models as well. And so what we did initially was to actually take our animals who've gone through that 10-day cocaine abstinence, which I mentioned to you before, and to look at their activity on the elevated plus maze. What we found is that if animals have gone through 10 days of cocaine abstinence, they actually show a decrease in open arm time and elevated plus maze, suggestive of an anxiogenic or an anxiety-producing effect. This is actually very similar to what we and many others in the field have observed with 21 days of chronic unpredictable stress. So again, you have an anxiogenic effect in the elevated plus maze. And so one of the things that we've tried to do is to see whether we can reverse this with specific manipulations of either the cholinergic system or the L-type calcium channels. And so here again, just showing you some of our earlier data, showing that if we block muscarinic receptors in the VTA, we can decrease cocaine-seeking behavior. It turns out that the same receptor blockade also reverses the anxiogenic effects of cocaine abstinence. So here we have saline animals that have about 45 seconds of open arm time. You can see that's lost during cocaine abstinence. If we block muscarinic receptors with scopolamine, we actually reverse that, and we see the animals are spending more time in the open arms again. So again, suggesting there are manipulations that can actually decrease craving can also alleviate potentially some of the anxiety-producing effects of the cocaine abstinence. And so we've started to look at that with our L-type calcium channels as well. So showing you some pilot data here. Basically, if we block L-type calcium channels in drug-naive animals, we have no change in open arm time. But in cocaine-abstinent animals, again, we see a dramatic increase in open arm time, again, suggesting that the L-type calcium channel blockade can reverse some of the anxiogenic effects of the cocaine abstinence. And just as the last bit of data before I close and leave it to questions, we've also, again, been looking at different types of models, so looking specifically at chronic unpredictable stress. This is actually a slide that I stole from a postgraduate in the lab, Narda Scobetti, who gave a very nice presentation to our molecular psychiatry group last week. But what she's highlighted here is just some of the different ways that we've tried to model some of these depressive-like responses in our animal models. And again, for this experiment, we're using chronic unpredictable stress, where the animals are exposed to various stressors over a 21-day period in a random order 2 times a day. And then we look at specific behaviors with the sucrose preference test, elevated plus maze, and also forced swim test. And what we found when we did those experiments is the following. So again, this is recently collected data that we're starting to put together, but looking at the sucrose preference test. So just to orient you, basically here showing in black all the animals that were home case controls, and gray all those that went through the chronic unpredictable stress. What we found was the main effect of the stressor itself, so there should a decrease in sucrose preference. We also have a main effect of isradipine, so we're giving our L-type calcium channel antagonist systemically, and we have an interaction between the two. So the CUS itself does indeed decrease sucrose preference test, and we're able to reverse that with the isradipine administration. We see a very similar thing in the females as well. So again, similar to what I showed with some of the cholinergic manipulations, suggesting that if we block some of these channels, we can actually reverse the effects of chronic unpredictable stress. We looked in the forced swim test as well. In this specific experiment, we didn't see any main effect of the stress itself, but we did have a main effect of isradipine. So we saw that isradipine did actually decrease immobility time, suggestive of an antidepressant-like effect, and also significant interaction with the stress as well. So if the animals had gone through stress, these isradipines seem to be that much more effective. Similar thing in the females as well. So again, suggesting that even after chronic stressors with some of the behavioral output, we can actually attenuate some of those with our isradipine manipulation that blocks L-type calcium channels. With the elevated plus maze, the data was a lot more variable, so it didn't pass the normality test, but we still did try to see whether there were any specific differences that did emerge. So there was no main effect of stress, but we did see a significant effect of these radipine itself. So suggesting that independent of whether the animals had been exposed to stress or not, is radipine did again increase their open arm time, suggestive of an anxiolytic effect, and we did have an interaction with the stress as well. At this point, nothing is pulled out with the females, but again, we can see quite a bit of variability. And so again, next steps here are really to start to actually use molecular tools to try and see whether there are differences of expression between the males and females of the L-type calcium channels, and also to see how stress may impact some of those L-type calcium channel expression levels as well. The third piece that we're trying to do is to actually see where these L-type calcium channels live within the VTA. There is extensive evidence that they do live on the dopamine cell bodies, but based on some of the work that I showed you before with our increases in dopamine, we're also going to use RNA-scope to see whether they may be in other cell types as well. And then again, just in terms of future directions, we're also going to start to look at some of the dopaminergic mechanisms in these responses to chronic stress, both to see how chronic stress impacts this phase of dopamine activity and how the L-type manipulation and muscarinic manipulation also impacts the dopaminergic activity and behavior. Again, with an eye towards translation, both for our L-type calcium channel work and also for our muscarinic manipulations as well. So again, just bringing it back to our working model, we've done a lot of work to really try and dissect some of these different components, but it seems that manipulations of both acetylcholine and L-type calcium channels within this VTA2 accumbens circuit do play an important role in both drug seeking and drug relapse behavior, and also in behaviors that we associate with mood disorders in terms of depression, anxiety, and hedonic related behavior, as well as motivational related processes. So with that, I'll get to the most important slide, which is thanking all of the people who've been involved in this work over the years. On the left, I've highlighted the current members of the Addy lab at Yale, Dr. Eric Nunez, who is an associate research scientist in the lab who performed some of the initial behavioral experiments with L-type calcium channel manipulations with a very talented post-bac student at the time, who is now a clinical psychology PhD student, Shannon Hughley. He's had a lot of other people who've come and contributed to this work over the years. Nardos Kabedi is the talented postgraduate who I mentioned, who's actually applying to graduate schools at the moment. So we'll be losing her soon, but happy to see her move on in several different ways. And then a host of other talented individuals in the lab who have contributed to this project and several other projects, which I wasn't able to talk about today. But very importantly, our collaborators on the L-type calcium channel work, specifically Anjali Rajadaksha at Weill Cornell, and then our collaborators at Vanderbilt University who have been working with us on the muscarinic receptor project and have given us great tools and lots of great insight along the way as we've been designing those experiments. So I think I did okay timing-wise, even with the Zoom blip in the middle, but I wanted to stop there and leave it open to questions.
Thank you so much. That was a great presentation. There are currently no questions in the Q&A, but they usually come a little bit late, so we'll give people a little bit of time to post their questions. But just a quick question, I guess the questions are coming in.
Okay.
So Claire is asking, I was super curious about the effect of isradipine on the sucrose preference task after chronic stress. How long does it take for the isradipine to take effect? And are these results after a single dose? Do you expect changes in the sucrose preference in animals where to take the isradipine over a longer period of time?
That's an excellent question. So to break it down a little bit, we haven't looked specifically at the timing yet to know how long it will take for this radipine to take effect. We had done that with some of our with our manipulations, specifically in the cocaine seeking. So I mean, if we translate that, for that it was about 15 minutes before we saw physiological effects in the brain. So I would suggest that's probably similar here as well. The test is long enough to be able to capture that. But to your second point, we have also been thinking about, in terms of translation, whether this is something that will have to be taken repeatedly. So we are also going to do experiments. These were all acute injections, but we're also going to do experiments with repeated injections to see if that has more efficacious effects and also if that has longer lasting effects. So we're thinking about that in terms of our comorbidity, whether we need to have daily isradipine or how long that will go for, and then how long after last isradipine will that effect last? As you can imagine, in a clinical setting, someone isn't going to take a medication right before they feel a craving come on or something like that, or even in a mood disorder. So things that we're actively thinking about. That's a great question.
Thank you. There are more questions from SORA. So there's a question about what do you think about endogenous changes in L-type calcium channel signaling according to different phase of addiction, for instance, drug seeking versus abstinence, and also what do you think about different distribution between two different types of the calcium channels, the calcium V1.2 and 1.3?
Yeah, another excellent, both excellent questions. So to the first, we have been thinking about that in particular as well. Actually, let me answer the second first. So we think that this is a CAV 1.3 specific mechanism in the VTA. The evidence from the Raj Dakshar lab and other places suggests it's CAV 1.3 in the VTA and CAV 1.2 in the nucleus accumbens, dependent on 1.3 actions in the VTA. So that's something that we're starting to look at. We may use shRNA to actually specifically try and isolate 1.3 in the VTA and see whether that is in fact the case. Actually, can you remind me of the first part of the question too? No, I've forgotten it. Oh, I think you're muted.
Sorry. What do you think about endogenous changes in L-type calcium channels signal according to different phases of addiction?
Yeah, so I didn't show all the data, but what we found is that the L-type calcium channel antagonism has no effect on cocaine taking itself. So it seems that at least endogenously, there's some type of plasticity that occurs during the cocaine abstinence phase specifically that's not engaged during cocaine taking. So that's something that we're going to look at, especially when we look at the expression, to see whether there's differential regulation during the abstinence period that then allows these things to emerge later on. So I don't have a clear sense yet of exactly what may be happening, but that's our hunch up to this point.
Thank you. Oh, one more question coming in. Are you looking for differences in behavior between males and females in drug responses? I'm also curious about anatomy. Is it known whether there are differences between male and females in these circuits? Or is it maybe hormonal?
Yeah, great questions again. So in terms of the behavior, there is evidence for differential behavior between males and females in terms of cocaine taking behavior and in terms of relapse behavior. So Wendy Lynch and others have done that work for quite a while. We haven't seen those differences emerge yet, but we haven't also done dose response analysis with our cocaine. So I suspect that those are also playing a role. In terms of the anatomy, I'm not aware of specific differences. We are also starting to think about differences in cycle as well. Our initial experiments were done in males. We're now in terms of the cocaine work. The mood disorder related work was done in males and females, but we also are starting to look at males and females in the cocaine related work. So some of that remains to be determined. My hunch is that there are sex differences that will emerge, and that may have to do with differences in receptor expression. I think we're starting to get a hint of that in some of our muscarinic receptor work with our pharmacology, but we're going to find out more about that when we actually do our receptor expression, which will be important because there may be different sensitivities in males and females based on the expression. And then from a therapeutic standpoint, that's something we would need to be aware of in order to make sure that whatever pharmacological intervention we use is appropriately dosed between males and females. So excellent questions. Thank
you. That's a great insight. We have one more question and a little bit of time. So this is probably going to be the last question. So sleep disturbance is also a common comorbidity for substance use disorders and mood disorders. So what are your thoughts about these acute and potentially future repeated pharmacological administrations on sleep?
That's a really good question as well. I don't have any specific insights there, but I think it's something that we definitely need to be thinking about and aware of. I mean, so I'll speak outside of the preclinical realm at the moment, but yes, I think that's definitely important for us to consider. And there is evidence for comorbidity there as well in some ways. I don't remember the exact prevalence, but Peter Morgan and others have actually looked at that and sleep disturbances in those who are navigating substance use disorders and how that actually impacts not only relapse, but also cognition as well. The interesting thing about some of that work is that sometimes it's not actually realized in those individuals that there are cognitive components that are coming into play unless you actually sit down and do a study. So, I mean, to your question, I think that's something we probably should be thinking about more so in our preclinical work as well. And I think moving forward, it would be important for us to think about whether any of these interventions that we come up with also have impacts on sleep, because that would tie back and also likely impact the effectiveness of the intervention in the first place. But that's a great question. I wish I had more, but that's going to force me to do some necessary homework. So I appreciate you asking.
One more question. We're going to try to add a little bit, and this is the last one. Katie is asking, LDT is a heterogeneous nucleus composed of cholinergic and GABAergic neurons. Does LDTG to VTA projections co-release acetylcholine with other neurotransmitters?
Another great question. I feel like I've said that each time. Not that I know of that I've seen, but I wouldn't be surprised if that is the case, because you're right. I mean, there's acetylcholine, glutamate, and GABA, all from the LDTG projecting to the VTA. So that's something that I think will be important for us to think about, especially again, if we're thinking about therapeutic pharmacological interventions. But I wouldn't be surprised.
Yeah, it's brain is a complicated system. Yeah.
Well said.
Well, it was, thank you so much for your talk. It was a great pleasure having you. Just a reminder to our listeners that our next seminar is going to be March 24th. Yeah, it was a great pleasure meeting you.
Thank you so much for hosting. Thanks for facilitating all the questions. And thanks to everyone for your patience with the unexpected Zoom freeze, but we made it work.
Yeah, current reality. Yeah. Thank you.